Studying the cell membrane and membrane proteins has a lot of potential for developing tools such as biosensors, for example. And one important design consideration is membrane composition, because membrane composition can in turn affect your membrane protein properties. And since protein expression and integration are sort of the first steps that can determine function and activity, I'm interested in learning how three different components, cholesterol, block copolymers, and lipid tail saturation can affect protein expression in vesicles. So the first component is cholesterol. It's naturally found in our cell membrane and has a very rigid ring structure. So when it's introduced into the membrane, it can affect one of the most important properties, which is fluidity. And that can in turn affect your membrane protein properties. The next one is diblock copolymers. They're tools that can be used to tune membrane properties synthetically. And previous studies have shown that membrane vesicles containing the block copolymer PEO, PBD, enhanced membrane protein folding. So basically, I'm interested in learning whether a short polymer versus a long polymer can affect expression. And lastly, we have lipid tail saturation. I'll be using DOPC and POPC. And the only difference is the amount of double bonds in each tail. And the double bonds become important when thinking about the intermolecular interactions between the tails themselves, which can affect your fluidity. So we take a cell-free protein synthesis approach. So within our reaction mixture, we have transcription and translation machinery provided by a kit called Pure Express. We provide DNA of a protein of interest, which is MSCL. It's an ion channel protein. And then we also provide the vesicles of varying compositions. And the way this works is the MSCL is fused to a GFP folding reporter. So we're assuming that if the protein is folded correctly, it will emit a fluorescence that we can read out on a plate reader. So first I'll go into the effects of cholesterol. So we make our vesicles of varying mole percent cholesterol, add that to our reaction mixture with our cell-free components and our DNA, allow that to run to completion, and we'll end up with these cholesterol vesicles with the embedded membrane proteins inside of them. So in the data here, I'm using fluorescence to compare expression. I'll refer to it as expression, but it essentially is the folding efficiency of the protein. So here we have DOPC, and we can see that as we increase the mole percent cholesterol, the expression levels also decrease, with the exception of this 10%. Next, we have POPC, and we can see that it kind of follows the same fashion. As we increase mole percent cholesterol, we see a steady decrease in expression as well. And if we want to compare them to determine the effects of lipid tail saturation, DOPC, which has one extra double bond and is more fluid, consistently has higher expression. Next, I'll move on to the block copolymers. We run these experiments in the exact same fashion. The only difference is I'm using a 0.95 kilodalton polymer, which I'll call the short polymer, and a 1.8, which I'll call the long polymer. So all of the data here is the same data, it's just represented in different ways for different comparisons. So here we can see that increasing the block copolymer molecular weight decreased our expression. So on the x-axis here, between 15% of the short polymer in DOPC and 15% of the long polymer, we can see that the long polymer resulted in lower expression. And we can see this throughout the data set. Whether it's 25% of the long versus short in DOPC, the long had less expression. And that is true for POPC as well. In the second section, we saw that increasing the mole percent polymer had little effect on expression. So at least between 15 and 25% of the short polymer in DOPC, we saw little change. 15 and 25 of the short polymer in POPC, little change. And that followed suit for the long polymer as well. And lastly, we saw that a higher degree of unsaturation or more double bonds in the lipid tails increased expression. And we also saw these same results with cholesterol as well. And so we can see on the x-axis, 15% of the short polymer, the only difference is the lipid type. Between POPC and DOPC, DOPC having more double bonds, we saw an increase in expression. And so we can see that whether it's 15 or 25% of the long or the short polymer, DOPC consistently had higher expression. So for future work, we can use lipids with higher degrees of unsaturation, different types of membrane components, other protein targets, and then we can move on to analyzing protein function and activity. And I would like to thank our collaborators at Northwestern, Zach Manzer, and Susan for all her help with this project.